See, the syllabus topic is describe the structure, functions and disorders of immunoglobulin. Okay, this is one part. Immunoglobulin, structure, function and disorder. And second part with a brief description of cellular and humoral immunity. This is another component. Basically, this involves entire immunology, and this basically is a nano form of immunology. The problem is that questions are not asked from this thing. One good example I will tell you. See, it is very difficult to cover this vast topic in just 2 classes. In your book, you will get only 4 pages, whatever book you read, and I will show you what is written in that book. You just, for your own information, I share something which are not available. See, this is immunology and this much is the topic, but as I told you, this is nano form, and you will be surprised that there is one big institute of almost 1/3 of this campus where National Institute of Immunology is there. It deals exclusively with immunology. It was established sometime in 1981 and then becomes functional from 1986. And during my MD period from 1988 to 1990, I visited this place several times because my work was, MD thesis related work was related to basically immunity. But it was a basically research institute. So you can imagine one institute is there and it cannot be covered. Second thing is that after you complete MD, there is a 3 years DM course on immunology. So that is the vastness of the subject. And all these super specialty hospitals, they are having an immunology department also, separate, dedicated. So just these are irrelevant things, but I told you just to make you aware of it that such type of institutes are available. This is one institute in our country, but in other places these are the things which are available. And again, many books and many journals are published every month on immunology. This will be basically a primer course for you to stimulate, to read and get you oriented about the subject. Now you see in the last supply examination, is 2026. There is one question of short note, classical pathway of complement activation. Actually, this is not covered in the small syllabus, but there is a short note of 5 marks. So you don't know what will appear in the examination. Very uncommon question, and this is a compulsory question also. This is a problem. Supplementary examination, such difficult question is set, which are expected for a 3rd prof examination during microbiology class. So we are helpless. You cannot blame me that we are setting very difficult questions. Second, there is one question, full question on what are the different components of innate immunity. How does it differ from adaptive immunity? Describe in brief the component of adaptive immunity. Full 10 marks question, compulsory. So, if you do not read properly and understand, you will not be able to answer. So, this is your favorite book, Jambulkar. I have deliberately copied it. He has made the entire subtopic in just 2 pages. and for you it will take only 15 to 20 minutes time, not more than that. There are several important points which are written, and I have gone through each and everything. Nothing wrong is written. Only thing that you have to understand, to memorize. Because Jambulgar books, there is no photograph. It is very difficult to remember, and very difficult to remember all the points. And this is under immunity. That is page number, the latest third edition, page number 409 and 4010. Today you definitely will go, after coming out from the class, you just read it. Second, under plasma protein, you will get another 1.5 page on immunoglobulin, structure and types, but there is nothing, disorder, no disorder is written. Just read these 4 pages, and whatever I am telling you today in the class, if you listen carefully, you will understand many things, and I want you to note down something which is not there in the notebook. Okay? Basically, if you want to know immunology, we must know what is an antigen and what is an antibody. These are very crucial part. These are the two main entities which involve immunology. So what I have decided that I will talk about antigen, what is an antigen, what is the definition of an antigen, and then innate and adaptive immune response, on which there is a question, and that you must understand also. Innate, the term innate means what we are born with that entity. মানে জন্মগত, at birth we acquire. It is present from the birth, since birth, that is innate. And adaptive means after we are born, then we are exposed to many things and we develop immunity, that is called adaptive immunity. And this adaptive immunity is having 2 components, one is cellular and one is humoral. That was the question. Third, the central role of T helper cell in immune reaction. Fourth one is self and non-self recognition, because our body immune system must recognize whether it is body's own tissue or it is a foreign one. Otherwise, if you cannot detect it, there will be disease. And these diseases are called autoimmune diseases. Antibody structure, types, functions, I will discuss tomorrow. Only you just read it today, so that whatever extra things I will tell you, you will note down. So, what is immunity? We very loosely use the term immunity. He doesn't have any immunity. Properly, he is suffering from cough, cold, this thing, that thing. And there are number of immunity boosting drugs which are available in the market, falsely claiming to increase the immunity, like Chyawanprash. But how do you measure immunity of any individual? How do you measure emotion? It cannot be quantified. But immunity can be quantified to some extent by measuring the antibody level of any specific substance, specific antibody, anti-polio antibody, anti-diphtheria antibody, anti-streptococcal antibody, specifically. Or we take the counts of the immunocompetent cells, T cells, B cells, which are immunocompetent cells. What is the count? If the count is low, that means there is a decreased immunity, and that does happen. So, basically immunity means state of being insusceptible or resistant to some noxious agent, usually pathogen. Pathogen means microbes in different forms, maybe bacteria, virus, or other microbes which you will read in your microbiology class. So, this is a state of being insusceptible, but I am not susceptible to infection. See, many of us are susceptible to infections. But if you look at the poor people who are exposed to too many bacteria, too many antigens, they don't suffer from that much disease. Like one example I am telling you, COVID outbreak. Too much precautions were advised, and many people wore many things. But if you go to the slums, they in a single room, 10 persons slept together. But there is no outbreak of COVID-19 in the slums area, crowded areas. Second, our body system gives immunity like our skin, hand, everything, body secretions. Now, a skin is an intact structure which is giving a physical barrier. Our hands, with that we use many things. And if we go on developing the habit of washing our hands frequently, then we are allowing it to be damaged, the entire structure. And those ladies who are very much fastidious about washing their hands and feet after every time, they get the entire area becomes moist and they do suffer from extensive fungal infection. So the natural immunity is destroyed. Repeated use of hand washers, hand sanitizers, this destroys the local immunity. That you remember. Don't try to get body clean very frequently because there are many things which are present, protective materials which are present on our skin, mucous membrane. If you wash it very frequently, they will be removed. Okay. So we acquire immunity naturally. Naturally means we are exposed to infection and from which we get immunity. One good example I am telling you, today in the one from a small family norm, all the mothers give the babies sterile water and water boiled and then it is given. So that doesn't allow intestinal immunity to develop. We have not taken that boiled water. We are exposed to infections. Those who are relatively lower socio-economic status, they are continuously exposed to the infections and they do develop immunity much better than others. Second, prior exposure means we have to get an exposure. This exposure may be a natural one or it can be given by vaccination, like immunization. During immunization, what is done? All of you have been immunized with polio, DPT, tetanus, measles, maybe hepatitis B also, where an antigen has been artificially introduced into your system, where the pathogenic part of that microbe is destroyed, but immunogenic part is retained to allow the immunity to develop. So immunity can occur by naturally, by prior exposure and by immunization. One thing naturally means we at the time of birth, we are born or endowed with some amount of immunity. That immunity I will tell you. That is called innate immunity. Three salient features. One is recognition of self from non-self. That is must, because our body must not mount any immune response against our own body tissue. And if it does, then it will cause an autoimmune disease. It might involve thyroid gland, it might involve pancreas, it might involve iris, it might involve skin. We do not know on what the body has mounted an immune response. And these are practically almost incurable diseases. And genetics are involved in it, and all immunological diseases do have a psychological component in it. Psychological upset, aggravation, fluctuate the course of the disease. These are not written properly in the book, but I have learned during my postdoctoral training in 2001-2002. Psychology, human psychology has a great deal to do with the immunity. That I will tell you later. Suppose one example I am giving you. In the Makar Sankranti day, too much cold is there. But people get exposed in the early morning beside the river. They take bath, remain in the cold water, but there is no outbreak of pneumonia on that particular day. No harm. But if the same lady or same person is exposed to take a cold bath in the house water, they will develop some cough, cold, because I have taken bath in the early morning when it is very much cold. So that one example I am telling you. So psychology has got a tremendous impact on the immunological issues also. On that day we are not getting infection. Other day we are getting infection. Specificity of the reaction, antigen antibody must be very specific. That specificity of the reaction. Third is memory of the response. Once I encounter any antigen, we must memorize it. Suppose we have encountered a bad person, we will memorize it that I have encountered a bad person. Now, next time when I meet that person, I will behave accordingly or with some kind of protection that, well, that person is a bad person, I have to deal accordingly. So, body develops mounts an immune memory also, that I will also discuss. So, types of immunity, this is written in the book, you need not copy, try to understand. One is innate immunity. Innate means what we are born with. What we are born with means we get the DNA from our parents, and these are germline genes. So, germline encoded genes are there in our system. With that we are born, and that confers innate immunity basically at the genetic level, germline genes. Second is acquired, means after we are born, we are having an acquired and passive immunity. Acquired means active immunity. What is active immunity? Means natural or it can be artificial. Active immunity. Suppose naturally we are exposed to an infection in any bacteria, because as a medical person, you will be exposed to many infectious patients in the ward, in the OPD. But all the healthcare professionals do not suffer from the disease. They mount an immune response. As a general person, you are exposed to the environment where many bacteria, many viruses are there. You will not develop disease. But though if you take an immunosuppressive drug, or if your condition is immunosuppression, like cancer, you will be required to take extra precaution not to be exposed to the infectious patient, because your body will not be able to mount any immune response. That you must know, that in immunosuppressed condition, that person has to be kept in isolation, or they have to take an extra precaution. So naturally we can get an antibody. This is having a two component, one is acquired, one is humoral. Humoral means related to body fluids, and it will be immunoglobulin, and another is cell mediated, which I will tell you. Artificial means antibodies developed in response to your vaccine. That's what artificially we have introduced, that antigen. Passive immunity. So, before I forget, I will write down. One is humoral immunity. One is cell mediated. This I will tell. That is the heart of today's discussion. So, artificial we can develop in response to vaccine. Passive immunity can be natural. We receive from the mother through the placenta. IgG crosses the placental barrier and gives immunity to the mother. And soon after birth, everybody takes mother's milk, and that confers immunity because it is having a natural immunoglobulin G, IgG, IgA that confers intestinal immunity. In addition, natural immunity, you will be surprised to know that during our early period of life, when we used to get an ocular infection, our mother used to put breast milk on the eyes. Breast milk is having some antibacterial, antimicrobial, antiviral substances. Similarly, almost all body tissue. We used to get cured of it. So, breast milk contains some antiseptic substances also. That is an important which gives an immunity to the baby. Of course, that is a passive because the baby is not developing actively. Passive, artificially, if the infection is very severe, body cannot mount any immune response, it is a dire urgent system, we give immunoglobulin directly in a high amount to the person. That is called passive immunity. Earlier, anti-immunoglobulin, actually immunoglobulin molecule was not available, so we used to get the antibody by raising an antibody titer in the form of serum in some horse. So that was or rabbit. That serum used to be given, but that serum used to cause some reactions. Later, with the advent of recombinant DNA technology, human immunoglobulins are available, and anti-tetanus serum, which was given during our time, is no longer given, but anti-tetanus immunoglobulin is given. The difference between active and passive, one good example I am telling you. So, you are a student, throughout the year you have studied every day and you have learned, acquired the knowledge, and that knowledge you are putting in the examination while writing the answer script. That is active. You have actively learned the entire subject and then you are responding to a basically question. Passive, you have not read anything, somebody has given a fully typed answer sheet to you and you are copying it you have copied it from that. That is what is passive immunity. So, active immunity will be long lasting, it will develop, it will take good amount of time to develop, but it will be memorized for a longer period of time. But in contrast, if you are provided with a passive immunity, that will be very much short lasting. Immediately after coming out from the examination hall, your memory is gone. Same thing will happen so long that immunoglobulin molecule will have a life, it will confer immunity. After that, it will not be able to give it. Is it clear? So, mother is giving because the baby could not mount any immune response. So, it is a natural process for survival. And active immunity we develop for our own survival after birth. So, there are three physical defense layer. One is physical barrier, tissues which are developed from the ectoderm. The skin, the entire skin is protecting as a boundary wall of the of our body. And the mucous membrane, if you take that skin, if you invaginate the skin into it, from the oral mucosa, it will go down to the anal cavity. So, it is basically invaginated. Similarly, if you look at the nostril, if you go inside, there is an epithelial tissue and it will cover the sinuses also. If you go through the nose and go down, again the bronchial tree, again it is covered with the epithelial cell. If you look at the urethra, if you go above, the entire thing is with epithelial. If you look at the vagina opening, that tract is also covered with epithelium, and the anal tissue, as I told you. So whatever is present inside, outside this thing, and as epithelium, that makes a physical barrier. That is a separate thing. But many persons also include it as, in many books also I have seen that it is also included as a part of innate immune system. Actually, after the physical barrier is just like a boundary wall. The Santiniketan Medical College is a human being, and we are having a boundary wall. This boundary wall prevents anyone from entering into the campus. If someone, a foreign material, a bad material enters into the campus, if by chance if it is near the gate, security will catch hold of it. But if it enters on the other side, it will not be able to do it. Our body tissue, which is there, suppose a foreign element is there, it has come on the surface. In the surface we are having an immunity, and it will not allow that particular foreign material to enter into the system. Now by chance if it enters, and if it remains within the tissue, in the actual intracellular matrix, then it will remain confined. It will be very easy to remove that particular substance. But if it enters directly into the blood, that means it is in the system. And if it is of larger size, then it might enter into the lacteals, lymphatic channels. And through the lymphatic channels, first it will come to the lymph node. Lymph node will try to destroy it, and it will definitely destroy in the majority of the cases. If it crosses that lymph node also, then it will come through the thoracic duct and right lymphatic duct to the main circulation, and it will cause an immunity to the systematic. Okay. So physical barrier, inner and skin is there. On the skin, don't think that skin is only the physical structure. Sebum is there. Several antibacterial substances are there. Antimicrobial substances which are present. So frequent washing the skin unnecessarily as an obsession will cause some amount of compromise with the immunity. Second, then comes the innate immunity, and then the adaptive system. Immunity, once something enters into the innate immunity system, innate immunity system is very rapid. It's just like a rapid action force. It will try to kill immediately. If it cannot, if the infection is so much, it will process, it will keep the infection to some extent at a halt and give time to the adaptive system to respond, which is responds a little late. So, although we have written all the things like innate immunity system and adaptive immunity system, they are separate things. They are independent, but they are interdependent also, and they act in tandem, means along with each other. In a family, a husband is a separate entity, wife is a separate entity, but they interact in tandem while in a family. That you must remember, but both are separate entities. It is something like that. In isolation, a family, good family cannot run with the husband alone or with the wife alone. That there must be an understanding, there must be cost of. But they are. So this is body's innate immune system. If pictorially I am showing it, so it is a large structure. This is the first line of defense. That also this exterior cover, which is a physical barrier basically. Physical barrier is different, I am telling you. That particular slide was taken from Immunology book, where these 3 layers are given, but commonly this is commonly understood that physical barrier is also included as a part of innate immune system. See, our body has evolved over crores of years of evolution. And during that evolution, we have encountered many bacteria, many virus, many microbes, and we have developed an innumerable number of receptors on our body. All the pathogenic elements which are present in our body, where our body cell like lymphocyte, this B lymphocyte basically gets converted into plasma cell, and that is a receptor on the B cell. Similarly, on the T cell there is a receptor. When the antigen enters into our body, it binds specifically with one type of lymphocyte that is having that receptor. This B lymphocyte is having almost each B lymphocyte about 1 lakh receptor that is in the form of immunoglobulin. So it will recognize that particular antigen. This I will tell you a little bit later. Let me complete this thing. Now, in the pathogen, there is one term called in immunity. Write down this thing: pathogen-associated molecular pattern. Means all the pathogens are having some characteristic feature. So they exhibit pathogen-associated molecular patterns. Means suppose each pathogen there is some kind of this thing or some kind of molecular pattern is there, and that is unique for their own survival. So this pathogen-associated molecular pattern, PAMP, it is written in your medicine book. It can also be called MAMP, microbe-associated molecular pattern, and that is unique for the microbe. So when a microbe enters into the body, that particular structure is there. Now, this is a highly conserved sequence of the gene. So, this is present in unique for all microbes, and it is vital for microbial survival, and they do not mutate, just like a consensus sequence. Now, how our body will recognize this thing? So, we are having a PRR. This is PAMP. This is pathogen-associated molecular pattern. Our body can recognize this thing. The immunocompetent cell can recognize in the innate system. So, they are having pattern recognition receptor. They call pattern recognition PRR, means pattern recognition receptor. Just note down this slide particularly. So, our body immune cell is having a structure like this. So, this is a pathogenic part, microbial part, and this is the host cell. So, PRR is present in the host cell, means our body cell, and this is a PAMP, this is present in the microbe. So, they interact with each other and recognize, and that's how they destroy this thing. Okay? So, pattern recognition receptor is a germline encoded genes, means we acquire it at the time of birth from our parents. So, that is one important component of innate immune system. Okay? And they are designed also, these are present in these macrophage, dendritic cell and neutrophils. Neutrophils, you know, is called as phagocytosis. Macrophage also causes phagocytosis, and dendritic cell also causes phagocytosis. So, they basically express on their surface this PRR. So, the moment they encounter an antigen, foreign material, they will interact and they will try to engulf it, destroy it. So, this is the pathogen-associated molecular pattern, this is pattern recognition receptor, and this can be present on the cell surface and also intercellularly also. So, that if the organism enters into the cell, again the cell there is a system of which will try to detect it. So now you see the skin structure, what will look like. So it is having a defensin and catalysin on its surface and antiside, just like milk, human milk. Human milk contains lysozyme. Human milk contains lactoferrin, which actually captures iron and bacteria cannot use it. It is having some oligosaccharide molecule of which gets coats the bacteria and prevents it from being pathogenic. So lysosome, lactoferrin and immunoglobulin A, secretory immunoglobulin A are present in almost all body fluids like tears, like breast milk, like mucosal secretions. So this body, apart from the physical structure, the secretions which are present on the mucous membrane and on the skin, they also confer immunity. And you see the phagocytes are there. This is called Lagrange cells. So by chance, if the foreign material enters into this by invading this superficial layer, phagocyte is there, that means these are basically police watchdog. They will kill. Now if it penetrates further, there is extensive mechanism of immune system which will take care of it. So adequate immune system is there physically. Now if you look at the mucous membrane, again the mucous membrane itself is a physical barrier, on top of which there are secretions which contains all antibacterial, antimicrobial substances. Okay. So just as I told you, so they will confer immunity, and again if it crosses the epithelium basal membrane, again all these immune system will come into the play. So what is immune system? I have told you, the adaptive immune system, this is basically the lymphoid system. Lymphoid system basically everything comes out from bone marrow, and then there is on the top you will get an tonsil. In the oral cavity, the adenoids at the posterior end of the nose, lymph nodes are acting as just like a police station in the entire body. The lymphatic vessels, thymus is there, spleen, PRRs patches, you know, in the intestine there are PRRs patches, and appendix also there is if you look at the histology, you will see that some lymphocytes are there, lymph nodes and the lymphatic vessels. This immune system which contains lymphocyte basically, they are responsible for the adaptive immunity. Now you try to understand this thing, this picture is there in your book, only PRR and PAMP is not written. First, innate, it is present from the birth. Very true. But how it is present from the birth? Because it is encoded by the germline genes. And germline genes we inherit from the parents. It is not present in the adaptive immunity. Adaptive immunity is acquired.
What is the opinion of
It is
the house?
after birth, it is not inherited.
Patient
Except placental
examination
transfer.
opinion নিতে হবে হাউ
So,
কিরকম opinion নিতে হবে লিখিত
this is part, not a
লিখে
part
দাও
of
আমি
adaptive
তোমাকে examination
immunity.
করব
And somatic selection, I will tell you this thing.
ভার্বাল
Depends
কনসেপ্ট
in
তার
the
মানে
first
সে
line
যখন
difference.
ঢুকছে তোমার কাছে একজন
Genetic
গাইনোকোলজিস্টের কাছে ইট ইজ ইমপ্লাইড
And this
যে
is
তার যে অসুবিধা
less
গাইনোকোলজির
potent,
প্রবলেম
but
সাধারণ বুদ্ধি
adaptive
যদি হয়
immunity is more
যদি
potent.
ইয়ে হয় তার গার্জিয়ান
you
থাকবে
mount
সঙ্গে,
an immune
যদি সে
response.
ইনসেন হয় বা কিছু
Specificity is
এবং
not
সে
specific.
তোমাকে বলবে
Because
যে
as you
আর
see,
ঢুকছে মানে
the
সে তোমার কনসেন্ট দিচ্ছে যে ইউ মে
molecular
এক্সামিন
pattern is a non-specific
আর
one.
কিন্তু ভার্বাল
But
কনসেন্ট নিতে গিয়ে অনেক সময় রিফিউজ
you
করতে
get exposed to
You should not go
diphtheria,
for that.
if you get exposed to streptococcus
না ডাক্তারবাবু এখন
infection, that is a bacteria,
বা আপনি দেখবেন না যে
specific antibody will be
থাকতে পারে
mounted against it.
চয়েস
Okay.
আমরা অনেক সময় বলি কি যাও ভাগো
Antibody
হ্যাঁ,
type,
ভাগ করে।
there is nothing
কিন্তু
and,
অনেক সময়
and
আমরা
no
দেখেছি,
antibody,
যখন
only
মাথা ঠান্ডা
cells
থাকে, থাকে,
actually
অনেক সময়
kill.
আমরা বুঝি-
So, design of the immune system, specificity, fine distinctions. If you look at the benzene ring, there is ortho, meta, para. And if you put an a functional group in the ortho position and try to develop an antibody, that antibody will not react with something which is with the same functional group present in the meta position or para position. Even that much small difference is recognized by the antibody. That is the specificity. Fine distinctions. Two quite similar molecules will not respond to that antibody. We need a very specific structure, having a very special arrangement at the molecular level. Ortho, meta, para can be detected by the antibody. So, you imagine the level of specificity, and diversity means we are having a broad repertoire of antigen. Since, as I told you, over course of years of evolution, our body has evolved. So, every B cell or T cell is having a receptor to recognize that particular foreign antigen. That is the broad repertoire, means storehouse of that antigen. Memory. Memory means initially when we get the infection, we will not be able to manage properly. But next time when we get the same infection, we mount an immune response and we will respond adequately, and that will be very rapid and that will be very much effective. Demobilization means what's the if there is an infection, suppose today there is a problem at the Bangladesh border. So our army or our security force will be deployed to that particular area, and they will try to control the invaders. Once that situation is controlled, neither the invader will be there, nor the security forces will be there. So they will be demobilized and restored back to their own position. That is what is immune. Similarly, immune system, immune cells will be, recruited at the time of actual infection, and then it will be cleared and distinguished from the self from non-self. Self means this is immunology. We develop immunological tolerance against our own body tissue. That is called an immunological tolerance. So antigen is a substance which provokes an immune response, immunological reactions, and produce antibodies against it. But antibodies are not only the issue; it mounts a immune response with or the T cell also. So specific facial I have told you. Suppose you know the blood group. Blood group, blood group A contains anti-B antibody. Blood group B is having an anti-B antibody. Okay. Now you cannot give blood group of A and to the blood group B because there will be reaction. You know, all of you know that O is an universal donor, which is partly true, partly false. And AB is an universal acceptor. See, AB doesn't have any antibody, I am telling you. This is not written in your book. O doesn't have any antibody, antigen on the surface of the RBC, but it is having anti-A and anti-B antibody in the serum. A group is an universal acceptor, it is having both the antigens, A and B, but no antibody. Now, if you take one drop of blood from AB group and one drop of blood from O group, and if you try to mix it, severe cross reaction will be there because the antigen is present in the AB group, antibody is present in the O group blood. So, what is done basically, the plasma is washed completely, and then the cells are suspended in normal saline and transfused, not the directly. Okay. So, N-acetyl galactosamine which is present in the A, B group will be basically D-galactose, so that will confer a separate type of antigen. So, a better definition is like this, that foreign or self molecule which are recognized by the adaptive immune system resulting in immune cell triggering T cell activation and B cell antibody production. This is a better definition but difficult to memorize. So, simple thing, at least you tell a substance which induces detectable immune response either humoral or cellular. That is the simplest definition of antigen. That will be the better definition. It will encompass everything. And if you write this definition, it will have a different altogether implication in the examiner's mind. He knows the entire thing total of the immune system. Antigen requires some properties like small molecular weight will not act. Some amount of molecular weight is present, so high molecular weight will be required. Protein and polysaccharides are good antigen. Foreign antigen, blood group antigens, all the tissues express different types of protein. See, liver of mine cannot be given to others. Liver, kidney cannot be donated by all persons to others. So we require tissue matching. The tissue must be matched, otherwise there will be rejection. So haptens are small molecules which by themselves cannot produce any antibody. But they react with the antigen. So allergen, many allergens we are allergic to it, they are all haptens. And in my immunoassay, you know that we have developed a progesterone immunoassay. Progesterone is having a molecular weight of 314. By itself, it will not be able to produce an antigen. So what I did, I coupled it with the bovine serum albumin, and then I injected onto the mouse. Okay, so that mounted an immune response. So epitope all the part of the antigen doesn't act as an antibody. Suppose this is the region which reacts with the antigen, that is called an epitope. And the entire immunoglobulin molecule doesn't react with that epitope. Corresponding part of the antibody is called paratope. This is the small part of the antigen which reacts with the antibody with the paratope region, that is an epitope, and its complementary part is paratope. So major histocompatibility complex, this is required for tissue matching, for tissue donation, particularly class 2 antigen. DR, DP, DQ, locus. You know the allogenic bone marrow transplantation, many things are told about bone marrow transplantation for thalassemia. This matching is required, HLA haplotype matching, HLA matching, you know what is that, what is done. This particular area is done. DR, DP, DQ, locus, whether it is matching or not. So finding a suitable donor is very difficult. Only a sibling can give, but that may not be 100% matching. So in a small family norm of 1 or 2 child, it is very difficult to get 100% identical haplotype donor. It is not possible. So some compromise will be there, and there is a chance of rejection also. So vaccine, basically how we take the vaccine is also we introduce infection. দেখো তো ইয়ে আছে কিনা ওই এসছে কিনা দেখো তো স্যার এসছে কিনা ছেড়ে দেবো। আচ্ছা দাঁড়াও। কমন ভ্যাকসিনস ইউ হ্যাভ টেকেন, একটা কথা খালি মনে রেখে দেবে, এই ফার্স্ট ভ্যাকসিনেশন হয়েছিল 1796-এ, ওই জেমস ফিলিপস নাম হয় না। ইনেট ইমিউনিটিটা পড়াতেই পারলাম না, বললাম অ্যাভেলেবল রেসপন্স ইমিডিয়েটলি টু ফার্স্ট ডেজে অ্যাকোয়ার্ড স্পেসিফিক চলে এসছে। আচ্ছা তাহলে ছেড়ে দিই, বলতেই পারলাম না কবে তো। ঠিক আছে, তাহলে একটু পরে ক্লাসটা নেবো। ওকে, ছাড়ছি তাহলে। এই সেলগুলো লিখে দেবে ইনেট ইমিউন সিস্টেমে, ওই প্যাটার্ন রেকগনিশন রিসেপ্টর অ্যান্ড পিএমপি। only thing is that these are the cells you know, natural killer cell only you may not know, monocyte macrophage you know, dendritic cell, epithelial cells এইটা আমি বলেছি অলরেডি। ওই পিএমপি মানে ডেঞ্জার সিগন্যাল চলে এসছে, এটা রিয়াক্ট করবে।